5,500 years ago, long before farming reached the forests of Siberia, children from the same hunter-gatherer families began dying together. In 2026, DNA recovered from their teeth revealed the killer and rewrote the known history of plague. But that was only the latest discovery. Across 10 ancient bodies and burial sites, scientists have uncovered a hidden history of disease stretching back thousands of years. All right, let's go. Number 10, the frozen man who carried a 5,000-year-old infection with no name. In September of 1991, two German hikers named Helmut and Erika Simon were descending from the Teisenjoch Pass high in the Ötztal Alps on the border between Austria and Italy when they noticed a shape emerging from the ice. It was brown, leathery, and unmistakably human. What they had found melting out of a glacier after more than 5,000 years was a man who would come to be known as Ötzi. He was the best preserved human body from that era ever recovered, complete with skin organs, clothing, and the copper axe he had carried on the day he died. For 2 decades, researchers focused on the outside of Ötzi, his tattoos, his tools, the arrowhead lodged in his shoulder that eventually killed him. Then, in 2012, a team led by Albert Zink, head of the Institute for Mummies and the Iceman at the European Academy of Bolzano published the Iceman's complete genome in the journal Nature Communications. Buried inside that genome alongside evidence that Ötzi had brown eyes type O blood and a genetic predisposition toward heart disease was something nobody expected: nearly 60% of the genetic sequence of Borrelia burgdorferi, the bacterium that causes Lyme disease. 60% of a 5,000-year-old bacterial genome recovered from a frozen man's hip bone is not a trace, it is a signature. It meant that a disease we associate with 20th century suburban backyards and deer ticks in New England had already reached a Copper Age herder in the Alps. That was 3,000 years before Rome was founded, and 5,000 years before anyone would understand what a bacterium even was. The discovery took a darker turn. Ötzi did not die of Lyme disease. He died of an arrow. The genetic evidence does not tell us how severe his infection was, or how long the bacterium had circulated in the region. What it does prove is that Borrelia burgdorferi was already infecting humans in Copper Age Europe more than 5,000 years ago. We like to imagine disease as something that arrives, a spillover, an outbreak, a moment we can point to and say, here, this is where it started. Ötzi's frozen body argues against that comfort. He was almost certainly not patient zero. He was one documented infection among an unknown number that may have come before him, none of which left a body cold enough, dry enough, or lucky enough to be found. For every Ötzi preserved by a glacier that happened to hold its shape for 53 centuries, there may have been many others who encountered the same bacterium and vanished completely, leaving nothing for us to sequence and nothing for us to name. Ötzi carried a flint dagger, a bow he never finished carving, and a remarkably well-preserved copper axe that reflected the advanced metalworking of his era. He also carried, without knowing it, a bacterium that would not be given a scientific name for another 5,000 years and would not be understood or treated until the modern era. He walked through the mountains infected with a disease science did not yet know existed, killed by a threat he could see carrying inside him, a threat he never could. Somewhere in the ice above the Ötztal Alps there may be others like him still waiting. We simply have not found them yet. Number 9, the liver disease that has been quietly inside us since before the wheel. In the early 2000s, archaeologists excavating a Stone Age settlement at Karsdorf in the Saxony-Anhalt region of Germany recovered the skeleton of a man who had died sometime between 25 and 30 years of age, roughly 7,000 years ago. There was nothing visibly unusual about his bones. He was by every outward measure an ordinary early farmer, one of thousands of skeletons pulled from Central European soil over 2 centuries of archaeology. What made him unusual was invisible, and it would stay invisible for another decade and a half. In 2018, a team led by Ben Krauss-Chiora, working with Johannes Krauss and other researchers, screened the Karsdorf man's remains for viral DNA. They also examined skeletons from 2 other German sites. Published in the journal eLife, their results identified 3 ancient genomes of the hepatitis B virus. The oldest from Karsdorf dated to roughly 7,000 years before present. It remains among the oldest human viral genomes ever recovered. 7,000 years is longer than the pyramids of Giza have existed by nearly 2,500 years. It is longer than the Sumerian city of Uruk, longer than the invention of the wheel, longer than writing itself. And the virus the researchers found was not a crude ancestor barely resembling its modern form. Its genomic structure closely matched hepatitis B, as it exists today, a virus that currently infects hundreds of millions of people worldwide and remains one of the leading causes of liver disease on the planet. At that point, conventional archaeology runs into a wall it cannot climb over. The ancient strains Kraus-Kiora's team found are not known ancestors of the hepatitis B circulating today. They represent distinct lineages more closely related to strains found in modern chimpanzees and gorillas than to any strain currently infecting humans. Those lineages have no close modern relatives and may have gone extinct. How and when later strains came to dominate remains unresolved. This is where the story stops being about one infected farmer and starts being about all of us. Hepatitis B is a blood-borne and sexually transmitted virus. For hepatitis B to persist in prehistoric populations, it required repeated chains of transmission through birth, blood, sexual contact, or other forms of exposure among people who had no concept of a virus and no way to see what was moving between them. Many people with European ancestry carry some genetic legacy from early farming populations, like the one to which the Karsdorf man belonged. What we do not know is how many of them died carrying this virus, how many outbreaks reshaped, which families survived to pass on their blood, or how many other pathogens rode alongside it, equally invisible, equally undocumented, equally erased, by 7,000 years of decomposition. The Karsdorf man was buried with no marker to suggest he mattered more than anyone else in his community. He did not know what was inside him, neither did anyone who buried him. It took 7 millennia, a molecular biology lab in northern Germany, and a question nobody thought to ask until very recently to reveal how viruses can evolve, disappear, and reappear in new forms across the long span of human history. Number 8. The children's disease that emerged as cities grew. In a museum storeroom in Berlin sits a small glass specimen jar catalogued as object BMM 655 from the year 1912. Inside is a fragment of lung tissue formalin-fixed, more than a century ago, taken from a 2-year-old girl who died of measles-related pneumonia in a Berlin hospital. For over 100 years, it was exactly what it appeared to be, an unremarkable relic of a disease that before vaccination killed children by the millions. Then in 2020, it became something else. A team led by Ariane Dukes of the Robert Koch Institute in Berlin extracted and sequenced the genetic material still preserved in that century-old tissue, hoping to answer a question that had frustrated virologists for decades: when did measles actually become a human disease? Published in the journal Science in June of 2020, their findings, generated using a technique called selection-aware molecular clock modeling, placed the divergence of measles virus from its closest relative, Rinderpest cattle disease, now eradicated worldwide, at approximately the 6th century BCE. The oldest written descriptions of measles date only to the 9th century CE, roughly 1400 years after that divergence date. Dux and her colleagues had pushed the biological origin of one of history's great childhood killers back by well over 1,000 years, beyond anything a historical document had recorded, using nothing but a jar of century-old lung tissue and the mathematics of viral mutation rates. That is where the discovery becomes genuinely unsettling. Measles cannot survive quietly in a small, isolated population. Unlike a slow infection that can smolder in a handful of families for generations, measles is explosively contagious and burns through susceptible hosts fast. Epidemiologists have estimated that sustaining it as an endemic human disease may require a population numbering in the hundreds of thousands or a closely connected network of settlements, large enough to keep the chain of transmission alive. A hunting band could not sustain measles. A small, isolated farming village probably could not sustain it either. Dense cities and connected populations changed that equation. Which means Dux's 6th century BCE estimate is not simply a date for a viral divergence. It may also preserve a biological fingerprint of growing urban populations. Somewhere around the time major Eurasian population centers were reaching the thresholds capable of sustaining this virus, an ancestor shared with the cattle disease Rinderpest, diverged into the human measles lineage. The genetic date does not identify the exact first human case, but it does suggest that expanding connected populations created the conditions in which a new human disease could endure. Every childhood killer with roots in animal domestication carries the same quiet implication. The intimacy we built with the animals we tamed gave us meat, milk, and labor. But that proximity also carried a biological cost that would not become clear for centuries, sometimes millennia. No surviving record tells us exactly when the first human cases appeared. But the viral divergence occurred in a world of growing, increasingly connected populations that would not understand what had happened for another 2,500 years. Only now, using the preserved lung tissue of a child who died in 1912, are we beginning to see the biological cost of the environments our ancestors built. Number 7, the world's oldest mummies and the parasite that outlived every one of them. In the early 1900s, the German archaeologist Max Uhle began excavating along a stretch of coastline near Arica in the far north of Chile at a beach that would later lend its name to an entire culture, Chinchorro. What Uhle found were mummies, not the accidental sun-dried remains that sometimes occur naturally in arid climates, but deliberately artificially preserved bodies, their organs removed, their skeletons reinforced with sticks and reeds, their skin repainted, and their faces covered with clay, masks. Radiocarbon dating would eventually reveal that the oldest of these mummies were created starting around 5000 BCE. That makes Chinchorro mummification older than Egyptian mummification by roughly 2,000 years. It was practiced by fishing and gathering communities along one of the driest coastlines on Earth, more than a millennium before Egypt built its first pyramid. For most of the 20th century, the Chinchorro mummies were studied primarily as funerary art remarkable for their craftsmanship and their age. Then, in 2004, a team led by Arthur C. Ofterheide of the Paleobiology Laboratory at the University of Minnesota Duluth published a study in the Proceedings of the National Academy of Sciences that looked past the elaborate exteriors and into the preserved tissue itself. Testing samples from 283 naturally and artificially mummified bodies, spanning roughly 9,000 years of continuous habitation along the Chilean and Peruvian coast. The team searched for a specific genetic signature, a segment of kinetoplast DNA unique to Trypanosoma cruzi, the parasite that causes Chagas disease. 41% of the tissue samples tested positive. Not a cluster, not a single outbreak. 41%, consistently, across 9,000 years, across 11 distinct cultural groups, regardless of the age, sex, or social status of the person being tested. Chagas disease, a parasitic infection that can silently damage the heart and digestive system over decades before killing its host, was not an occasional visitor to this stretch of coastline. It was essentially a constant. The comforting version of this story falls apart here. The infection rate did not decline as human communities grew more sophisticated, built more permanent structures, or developed the kind of social organization we associate with advancing civilization. It stayed roughly flat for 9,000 years, through the earliest Chinchorro fisher-gatherers, through the agricultural societies that followed them, through the more elaborate settlements that came later still. Ofterheide's team concluded that the parasite's wild transmission cycle passed among rodents, opossums, and other mammals through a blood-feeding insect vector was already fully established along this coast before the first humans ever arrived. The earliest Chinchorro people did not introduce Chagas disease to this landscape. They walked into a disease ecosystem that had been running for an unknown span of time before them and simply became one more mammalian host among many. This is the part conventional histories of civilization tend to leave out. We tell the story of the Chinchorro through their achievements, the world's oldest known deliberate mummification tradition, and a sophisticated ritual response to death predating Egypt by 2 millennia. It was created by people the wider world barely remembers today, while museum culture still lavishes far more attention on pharaohs than on the fisher-gatherers of the Atacama coast. What the tissue samples reveal is a second, quieter narrative running underneath the first. While these people were perfecting one of the most elaborate mortuary traditions in human history, roughly 41% of the individuals tested carried the parasite's genetic signature. The parasite had arrived on this coastline before they had and would still be there in new hosts, long after their meticulously preserved bodies were found. Number 6, the pit of butchered bones that hid a second, quieter killer. Between 1,972 and 1,976 archaeologists, excavating in the Mendip Hills of Somerset, England, were searching for the entrance to an underground cave system when they found something else, entirely a natural limestone shaft, plunging 15 meters into the earth filled with human bone. The site was called Charterhouse Warren, and for decades the scale of what lay inside it was not fully understood. It took until 2024 for a team led by Rick Schulting of the University of Oxford to reveal the full extent of what had happened there roughly 4,000 years ago. Published in the journal Antiquity, the study described the largest known act of interpersonal violence in prehistoric British archaeology. Yet the site remains largely unfamiliar outside specialist circles. Schulting's team examined more than 3,000 individual bones and fragments representing at least 37 men, women, and children. What they documented was not a burial. Cut marks consistent with dismemberment and defleshing covered the remains. Skulls showed blunt force trauma. Long bones had been fractured around the time of death, and some bore marks consistent with human chewing suggesting the bodies had been partially cannibalized, not for sustenance, since abundant cattle remains were found alongside them, but the researchers proposed as a deliberate act of degradation. The surviving remains preserve no clear evidence of a defensive fight. Whoever these people were, they appear to have been taken by surprise, killed, and butchered. That on its own would be disturbing enough. A second investigation complicated the case. In 2023, an entirely separate team led by Pooja Swally of the Francis Crick Institute in London and published in Nature Communications was screening ancient DNA from British Bronze Age skeletons for evidence of Yersinia pestis, the bacterium behind plague. Among the individuals whose remains tested positive, representing the earliest confirmed evidence of plague in Britain roughly 4,000 years old, were two people recovered from Charterhouse Warren, not a nearby site. Not a related community, the same shaft. The same pile of dismembered bone that Schulting's team had spent years documenting as a massacre. Two entirely different investigations using two entirely different methods converged on the same 15-meter shaft and found two entirely different horrors layered on top of each other. Neither study was designed to explain the other. Schulting's violence research makes no claim about disease. Swally's plague research makes no claim about the massacre. And so a question sits between the two papers, that neither one answers were these people killed because of what had already begun spreading among them. Did fear of a contagion nobody could name or treat curdle into violence against the people already carrying it? Or is the presence of plague in that shaft simply a coincidence? Two unrelated tragedies that happened 4,000 years apart from our vantage point, but mere moments apart from theirs, to end up in the same hole in the ground. Nobody currently knows. The researchers found that the explanations we reach for instinctively resource competition, ethnic conflict, or climate stress did not convincingly fit the available evidence. There is no genetic evidence of a distinct rival population moving into the area. There is only a community apparently attacked with a ferocity that left no one able to defend themselves, some of whom it now turns out were also infected with one of the deadliest bacteria in human history. We are left with an uncomfortable arithmetic. A shaft. 37 bodies. 3,000 cut, broken, and gnawed bones. Two confirmed cases of plague sitting among them unremarked upon for 4,000 years until a molecular lab flagged them almost by accident. Whatever happened in that community in the centuries around 2,000, 200 BCE, whether disease came first, violence came first, or the two arrived together in some sequence. We may never reconstruct the people at Charterhouse. Warren were failed. Twice. Once by whoever did this to them. And once by 4,000 years of silence, that very nearly kept us from finding out there might have been two killers in that pit, not one. Number 5, the plague that helped break a golden age and still doesn't have a confirmed name. In 1994, ahead of construction on a new line for the Athens metro archaeologist Efi Baziotopoulou Valavani of the third effort of prehistoric and classical antiquities began a rescue excavation just outside the ancient Kerameikos cemetery, the primary burial ground of classical Athens. What her team uncovered was a shaft containing roughly 90 skeletons, among them at least 10 children, thrown in without the usual soil layers separating individual burials. Scattered among the bodies were around 30 small, cheap ceramic vessels, a burial offering wildly disproportionate to the number of dead dated to between 430 and 420 BCE. Baziotopoulou Valavani later described the grave as lacking any monumental character whatsoever. The bodies, she concluded, had been placed in the pit within a day or two of death. That date and that haste matched something that had been sitting in the historical record for over 2,000 years, the Plague of Athens. The historian Thucydides survived the outbreak and described it firsthand. He recorded a city descending into panic as bodies piled up faster than proper burial rites could accommodate them. Thucydides estimated the epidemic killed roughly a third of the population and claimed the life of Pericles, the statesman who had led Athens into its golden age. But Thucydides, meticulous as he was, could not identify the disease itself. His description has since been used to argue for smallpox, typhus, measles, and more than 24 other candidates. In 2006, a team led by Manolis Papagrigorakis of the University of Athens attempted to settle the question using dental pulp extracted from 3 teeth recovered at the Kerameikos site testing for 6 known epidemic pathogens, including plague, typhus, and anthrax. All 6 tests came back negative. On a 7th attempt, the team identified DNA sequences resembling Salmonella enterica serovar, typhi, the bacterium that causes typhoid fever, and published their conclusion in the International Journal of Infectious Diseases, naming typhoid as the probable cause of an epidemic that had haunted historians for 2.5 millennia. The proposed diagnosis soon began to fall apart again. Within months, other researchers pointed out that the recovered sequences diverged from modern typhoid strains by a margin large enough to cast real doubt on the identification, and ancient DNA extracted from 2,400-year-old dental pulp is notoriously vulnerable to contamination and degradation. The typhoid diagnosis has never been fully accepted, and the debate did not end there. As recently as 2026, researchers re-examining the case pointed to a detail in Thucydides' own account that the typhoid theory cannot explain. He specifically reported that dogs, birds, and other animals scavenging the corpses also died. Typhoid fever does not typically kill animals that merely come into contact with infected human remains. That single observation made by a man standing in his own stricken city 24 centuries ago contributed to a different proposal published in 2026 that an unusually invasive, non-typhoidal strain of Salmonella may offer a better explanation. It is a new interpretation, not a confirmed diagnosis, and the true cause of the epidemic remains unknown. Among the roughly 10 children recovered from the pit, one has since been given a name she was never called in life: Myrtis. She was an 11-year-old girl whose skull survived intact enough to allow a forensic facial reconstruction. Her reconstructed face was later put on public display in Athens, giving the epidemic's youngest victims a presence in the record they never had in death. She has no stake in typhoid's critics or its defenders. She is simply 11, permanently one of roughly 90 people who did not receive the burial their city would have given them in any other year. 2,500 years after it happened, using dental pulp, ceramic dating, statistical DNA analysis, and the surviving words of an eyewitness who watched his own neighbors die, we still cannot say with certainty what killed so many people in one of history's most consequential cities. Somewhere beneath a metro station in modern Athens, commuters walk each day directly above the spot where a civilization's confidence in its own permanence was buried in haste by people who had run out of time to do anything else. The disease has outlasted more than 24 proposed diagnoses. It may outlast the newest one as well. Number 4, the pandemic that shook an empire and then vanished from the genetic record. In the Aschheim-Bajuerenring cemetery outside Munich, Germany, lies the grave of a man archaeologists now know only as a 120, buried alongside 2 other individuals, sometime between 525 and 550 CE, a date fixed by the NRA's Dacom, grave goods left with him. For decades after the grave's excavation, a 120 was simply one skeleton among thousands recovered from early medieval Bavaria. Then, in 2014, a team led by David Wagner of Northern Arizona University, working with researchers including Michaela Harbeck and collaborators at McMaster University's ancient DNA facility, extracted DNA from his teeth, along with the teeth of a second individual buried nearby, known as A79. Published in the Lancet Infectious Diseases, their genomic analysis confirmed what smaller studies had suggested a decade earlier. Both men had died, carrying Yersinia pestis, the bacterium behind bubonic plague. What Wagner's team had found was direct genetic evidence for the plague of Justinian, the first of 3 pandemics. Yersinia pestis is known to have caused in human history, named for the Byzantine emperor whose empire it devastated. The contemporary historian Procopius, writing from within Constantinople as the outbreak unfolded, described a city overwhelmed by the dead, its normal burial grounds, exhausted corpses stacked inside the towers of the city walls because there was nowhere else left to put them. Some modern estimates place mortality in the hardest hit areas as high as 40%, although the pandemic's overall demographic impact remains debated. Sequencing identified 66 unique chromosomal and plasmid variants in the two reconstructed genomes. 23 altered protein sequences, but only two occurred in well-characterized virulence factors. The genomic evidence challenged a story historians had been telling for decades. The long-standing assumption was that the plague of Justinian and the Black Death, which would devastate Europe 800 years later, were part of a single, continuous chain-one pandemic, smoldering somewhere before flaring up again, centuries afterward. Wagner's analysis proved otherwise. The Justinianic strain sits on its own branch of the Yersinia pestis family tree, closely related to strains found in modern rodent populations in Asia, and it has no known descendants. Its lineage appears to have gone extinct. Whatever caused the plague of Justinian to disappear, after roughly 2 centuries of recurring outbreaks, that particular branch became a genetic dead end, which means the Black Death was not a resurgence. It was a second pandemic caused by a different lineage of the same bacterial species emerging from a wild reservoir centuries after the first pandemic lineage had run its course. Had that Bavarian lineage persisted instead of dying out, the medieval world that followed might have taken a shape we would not recognize at all. Different families might have survived to have children, and different bloodlines might have inherited a Europe reshaped by recurring plague. Instead, the first pandemic lineage disappeared from the known genetic record. Instead, two of history's most devastating pandemics, separated by roughly the span between the fall of Rome and the invention of the printing press, were not chapters in the same story. They were separate catastrophes, caused by different branches of Yersinia pestis, whose precise animal reservoirs and routes into human populations remain under investigation. This is the detail that should unsettle anyone comforted by the idea that a pandemic once survived is a closed chapter. The first pandemic lineage disappeared from the record, but Yersinia pestis persisted in other wild reservoirs and lineages. Centuries later, under conditions still not fully understood, a different branch spilled into human populations and produced the Black Death. The plague of Justinian severely damaged communities across the Mediterranean, although historians still debate how far it weakened the Byzantine Empire as a whole. What the evidence does show is that one pandemic lineage can disappear while the larger microbial threat remains. Number 3. The farming community plague returned to 3 times. In 2001, archaeologist Carl Guran, Sjogren of the University of Gothenburg, excavated a Neolithic passage grave at Frelsegården in the Falbygden region of Sweden and found something that did not fit the usual pattern of Stone Age burial. Instead of a small number of individuals, the tomb held the remains of at least 78 people. The burial pattern initially suggested an unusually concentrated period of use, although later genomic work would reveal multiple generations and later reuse of the chamber. For 17 years, that fact sat in the archaeological record as a puzzle without an obvious answer. Then, in 2018, biologist Nicholas Raskovan, working alongside Sjogren and geneticist Simon Rasmussen, screened ancient DNA from the Frelsegården skeletons for evidence of infectious disease. In the remains of a 20-year-old woman, he found something unexpected. It was a strain of Yersinia pestis so genetically basal and so close to the root of the plague family tree that it predated every other strain known at the time. Published in the journal Cell, the team named it GOC2 and dated it to between roughly 5040 and 4867 years before present. A second individual at the same site, a young man of similar age, carried traces of the same strain. Raskovan's team proposed that a prehistoric epidemic may have spread along the expanding trade networks, connecting late Neolithic Europe. It might help explain the concentrated burials at Frelsegården and something far larger, the Neolithic decline. That was a sharp, still-debated population collapse recorded across large parts of Europe between roughly 5300 and 4900 years ago. The scale comparison here is almost impossible to hold in the mind at once. Raskovan's molecular clock analysis suggested that at least 3 genetically distinct lineages of plague, GOC2 among them, were already circulating across Eurasia within the same few centuries, thousands of years before anyone would build a pyramid, invent a wheel, or put a word to paper. A disease we associate with medieval Europe, convulsing under the Black Death, was already diversifying into multiple branches, while Stone Age farmers in Sweden were still burying their dead with flint tools. Mainstream skepticism arrived quickly. In 2021, a separate research team examining an even older, independently discovered strain of plague, concluded that the genetic evidence did not support a prehistoric pandemic at all, arguing instead that early Yersinia pestis infections looked more like isolated one-off spillovers from animals than a raging transmissible epidemic. For several years, whether plague truly caused the Neolithic decline or merely happened to be present alongside it, remained genuinely unresolved, argued back and forth across competing papers. Then in 2024, a team led by Fredrik Valur Searsholm at the University of Copenhagen, again working with Sjögren, returned to Sweden with far more data than Raskovan's team had available 6 years earlier. Analyzing ancient genomes from 108 individuals recovered from 8 separate megalithic graves and a stone burial cyst, the team found Yersinia pestis in at least 17% of the population sampled. They reconstructed 4 multi-generational pedigrees. The largest included 38 sampled individuals, from a family spanning 6 generations. The infection did not appear once. It appeared in 3 distinct infection events within the same Neolithic community over a period of roughly 120 years. 17%, 3 waves, 6 generations, a multi-generational community. This was not a solitary infected woman anymore. This was documented, repeated infection moving through related families over more than a century in a community with no concept of quarantine, no understanding of contagion, and no way to know that the same pathogen had returned a second and third time. The skepticism raised in 2021 was answered with better data, more samples, and a clearer picture of a community repeatedly exposed to something it could never see coming and never see leave. We do not know exactly how many of these infected people died because of plague, but we can imagine what repeated infection meant inside that community, one generation burying its dead, followed years later by another generation facing the same invisible pathogen. Gordon was not one tragedy. It was at least 3 layered across 6 generations in a farming village that thought each time it had already survived the worst of what was coming for them. Number 2, the loneliest death in the history of the plague, and for 6 years the oldest one ever found. Sometime in the 19th century, 2 human skeletons were excavated from a low, unassuming shell, midden beside the Salika River at Rynjukälns in what is now Latvia. The remains passed into the collection of the German pathologist Rudolf Virchow in Berlin, and then for reasons no record fully explains, they simply disappeared. For over a century, they were presumed lost. In 2011, they resurfaced, rediscovered inside the archives of the Berlin Society of Anthropology, Ethnology, and Prehistory, exactly where they had been sitting the entire time unexamined, while an entire century of scientific progress happened without them. Once rediscovered, archaeologists returned to Rynjukälns and excavated 2 additional burials, bringing the total to 4 individuals, likely members of the same small community of hunter-fisher-gatherers. In 2021, a team led by Ben Kraus-Chiora, head of the Ancient DNA Laboratory at the University of Kiel, and Julian Sussat, screened all 4 for bacterial and viral pathogens. In the remains of one man, a 20 to 30-year-old individual, the team designated RV 2039, they found something unexpected, a nearly complete genome of Yersinia pestis. Published in the journal Cell Reports, radiocarbon dating placed his death between roughly 5300 and 5050 years ago. Phylogenetic analysis placed his infection near the root of the plague family tree. The lineage emerged not long after Yersinia pestis diverged from its less dangerous bacterial ancestor. It was at the time the oldest confirmed case of plague ever found in a human being, pushing the pathogen's documented presence back roughly 2,000 years beyond what had been established just 6 years before. The team's own description of the finding placed this early strain remarkably close to the organism's actual genetic origin, closer than anyone studying the disease had ever managed to get. This discovery pivots away from everything the story has told us so far. Every instinct built up by the previous entries on this list points toward pandemic, toward mass graves, toward whole communities and bloodlines collapsing together. RV 2039 offers something different and, in its own way, more disquieting. His bacterial load was unusually high, a pattern that in modern rodent studies correlates with a less aggressive, more localized infection, rather than an explosively contagious one. He was buried carefully in the same manner as everyone else at the site. The people buried around him were not infected. There is no evidence of a wider outbreak, no second grave, no cluster of the dead. Kraus-Kiora's team concluded that RV 2039 probably contracted the infection directly from an animal. A beaver was one possibility because beaver remains were common at the site, and the animals can carry related bacteria. The evidence at Renucalns point to an isolated case rather than a local outbreak. Their published conclusion explicitly stated that the data did not support the kind of prehistoric pandemic that other researchers studying the Swedish farming village at Frelsegården had proposed just 3 years earlier. There is a particular strangeness to how long RV 2039 waited to be understood, and how many times. He was buried beside the Salaka River for roughly 5,000 years before anyone dug him up. Then, once excavated, he was buried again, this time inside a 19th century museum archive catalogued and set aside indistinguishable from thousands of other unremarkable specimens for more than a century, while the science capable of finally reading what was inside him did not yet exist. Renucalns did not need a natural disaster or a bureaucratic accident to erase this man's story a second time. It only needed ordinary institutional neglect, the same quiet drift into irrelevance that swallows most archaeological collections everywhere, and very nearly swallowed his completely. Here, then, was plague as close to its genetic origin as researchers had reached in 2021. Not a catastrophe, not a collapse, not a bloodline erased across generations, but one man alone in a small hunting community beside a Baltic river. This early strain had not yet acquired the genetic machinery associated with efficient flea-borne transmission, which would appear roughly 16 or 17 centuries later. At Renucalns, plague looked more like isolated bad luck than apocalypse. And yet, the organism inside RV 2039 and the organism that would eventually kill perhaps half of medieval Europe are genetically the same species separated only by time and a handful of mutations. Somewhere in the gap between this solitary hunter's death and the mass graves that would eventually follow an ordinary zoonotic accident became the deadliest pathogen in recorded history. Nobody knows exactly where that transformation happened or how many times it had to happen quietly in unmarked graves. Nobody has found yet before it finally succeeded. Number 1, the oldest known plague outbreaks and the 5,500 years they spent waiting to be found. Around the shores of Lake Baikal in the forests of southeastern Siberia, archaeologists have spent decades excavating the cemeteries of hunter-gatherer communities who lived, fished, and died along its coastline thousands of years before agriculture ever reached the region. 4 of those cemeteries, their burials meticulously recorded and their skeletal remains preserved in regional archives, became the subject of an international genomic study published in the journal Nature in June of 2026. The research was led by Ruarid MacLeod and Frederick Searsholm, with senior researchers Martin Sikora and Eske Willerslev. Extracting DNA from the teeth of the dead, the team searched for a single organism, Yersinia pestis. What they found rewrote the opening chapter of this entire story. Across the 4 cemeteries, 39% of the individuals tested carried genetic evidence of plague infection. Reconstructing family relationships from the DNA itself, the researchers determined that the disease had moved through small related kin groups in a pattern consistent with transmission between people, not merely isolated spillovers from animals. The outbreaks occurred in 2 separate phases, centuries apart, the earliest beginning approximately 5,500 years ago. That is several hundred years earlier than the Latvian hunter-gatherer known as RV 2039, several hundred years earlier than the Swedish farmer known as Gok 2, and as of this recording, the oldest confirmed evidence of plague ever found anywhere on Earth. The likely animal source was not the black rat that would carry the Black Death across medieval Europe 3,500 years later. The researchers proposed that it was probably the marmot, a large burrowing rodent that lived alongside these communities, and that they hunted extensively and a natural reservoir for Yersinia pestis in parts of the world today. There were no cities at Lake Baikal 5,500 years ago, no granaries drawing in rats. None of the conditions researchers have long assumed were necessary for plague to become genuinely dangerous. There was only a lake, a handful of extended families, the animals they depended on to survive, and a bacterium that did not wait for civilization to give it permission to become lethal. Agriculture had already transformed other parts of the world, but it had not reached these hunter-gatherer communities. Large cities were not required here. The pattern is consistent with transmission between people inside small related groups beside a frozen lake. Plague did not need civilization's permission to become lethal. At Lake Baikal, the human cost becomes almost unbearable to sit with. The infections were not gentle. Reconstructing the kinship pedigrees at the sites, the team found that the first outbreak swept through an affected family within a single generation, and that mortality was acute, falling with particular severity on children between the ages of 8 and 11, not elderly individuals already weakened by age. Children, specifically at exactly the age when they would have been old enough to help with the hunting and fishing that sustained their families, and young enough that their deaths would have gutted whatever future that family had imagined for itself. This is where every earlier entry on this list collapses into a single, unbroken line. Across 9 other discoveries, we have watched dangerous pathogens cross into human beings and persist for thousands of years on several continents. They left behind infected bodies in Somerset, repeated plague cases in Sweden, devastated communities around the Byzantine Mediterranean, and a city's golden age buried beneath what is now a Greek subway platform. Lake Baikal shows us the earliest chapter of that catastrophe discovered so far. It begins not with an empire, not with a city, and not with the domestication of a single animal. It begins with hunter-gatherer families losing their children to a disease, with no name, no cure, and no explanation any of them could have reached for. If the death of a single hunter beside a Latvian river represents plague at its loneliest, then the outbreaks at Lake Baikal represent something worse, proof that isolation was never guaranteed. A zoonotic spillover followed by transmission within a small related group may have been enough. The same bacterial species would one day travel the length of the Silk Road, sail on merchant ships into Mediterranean ports, and kill perhaps half of Europe. But 55 centuries ago, it was already capable of spreading through Siberian hunter-gatherer families without agriculture, writing, or cities to give it a larger stage. You are here reading or listening to this because somewhere in the unbroken chain of ancestors, stretching back before Lake Baikal, before Rhinukolns, before Frelsegården, every single one of your direct forebears survived. Not most of them. Everyone without exception. They survived across tens of thousands of years and an unknown number of outbreaks, including events buried in places we have not yet found and involving organisms we have not yet identified. Many of those outbreaks may have taken children, like the ones at Lake Baikal, like Myrtis in Athens, and like the victims whose names we will never know. This particular chapter of the story took 5,500 years to surface. It surfaced only months before this was written. There is no reason to assume it is the last one still buried, and no reason to assume the ground has finished giving up its dead. If you want to see more videos like this, click the video on screen now and make sure to subscribe.